All righty. Our next featured session is Contemporary Trials and Next Steps, and will take us to lunch. This session will feature updates on trials from 7 different organizations. There will be a Q&A at the end of this session if we have time. And be sure to stay to the end because I have a very special guest joining me to send us off to lunch. I'm not going to tell you who it is right now. So without further ado, please welcome Dr. Cristina Sampaio, Chief Medical Officer from CHDI, to highlight the critical issues in HD trials. Following her introduction, Dr. Blair Levitt will moderate the remainder of the session. Dr. Sampaio, come on up.
Good morning, everyone. Thank you very much to Daniel and the organizers for inviting me. The time is short, so I'll try to rush through a number of important topics related with clinical trials. That's how I organized my talk. The main topics are these 3 that I listed here, namely selection of the target population, choice of outcomes, duration of follow-up. I think I met every single company that is in this room, and the ones that are not. We are always going over and over these topics. And sometimes it seems a déjà vu, but there is always something new. Before I go in depth on these issues, I want to briefly mention something that is in the mind of everyone. So every single company is praying to different gods to get accelerated approval. And the issue is that indeed accelerated approval from FDA, that by the way is not the term, neither the process that exists in Europe, might be indeed a solution, or at least a short-term solution. But I want to raise some caveats. I just said that I agree that it's very likely that the path forward to see the first drugs in the market might be through accelerated approval. But I wanted to bring some cold water to this enthusiasm in the sense that there has been a lot of progress on approvals of drugs for cancer in the last decades. Many of them went through accelerated approval, and only about half of them were indeed confirmed as having an impact on survival and quality of life. So when they were approved on shrinking of the tumor, for example, which is the most common intermediate biomarker that is used for accelerated approval, then these drugs didn't go on to confirm in the latest trials, and many of them were withdrawn from the market, others weren't, and that's another discussion. On the other side of this slide is another type of data that says that accelerated approval indeed was meaningful for about 1 million people, and that they contributed for 250,000 years life gained. So it's a half full versus half empty kind of glass. Accelerated approval brings value to patients, but it also creates a burden to the society, and it's important to balance these uncertainties. So I'm just saying that many people think that just because a drug gets approved through accelerated approval, or even sometimes full approval, that this means that is the end of the story, and it is not. So with this caution, I'm going back to the basics. So first of all, yesterday we heard, maybe even in a certain negative tone, that there are drugs that improve and drugs that slow the disease progression. We have some of the ones that improve. We don't have yet any of the ones that slow the disease progression. But these are 2 different phenomena. Both of them are important for the patients. Both of them are relevant. But it's true that the patients it's easier for the patients to recognize improvement. than it is for them to recognize slow of the duration. It's almost impossible that a person recognizes, oh, yesterday or months before or last year, I was progressing much more faster, and now I slow down. You know, they can recognize a car slowing down, but they don't recognize the disease slowing down. And that's a big challenge for demonstration of clinical relevance. This is the integrated staging system that we developed for purposes of doing research and for the purposes of facilitating clinical trials. We heard several commentaries already about how to transfer this to medical care. We are not ready for that. This is a process to be used in research. But it is very important to note that we are not debating the diagnosis of the disease using the HDISS. The diagnosis is made by a positive test. A positive test of 40 or more, because it's the only range where the disease is fully penetrant. Before 40, we are still working in this process. So this only applies for 40 or more. But this is how we select populations for clinical trials. And this brings a lot of advantage that I cannot run completely through today, but that's an important aspect. Another important thing is that HD has a very special flavor when it comes to the concept of rare disease. It's almost, let's say, an easy process to go to FDA or to EMA and get orphan drug designation for a drug targeting HD, because it meets the criteria for low prevalence. But low prevalence is defined in the case of US as less than 200,000 people affected by the disease. And it comes with this, that most of you quote a number that is, there are 30,000 people with HD in USA. And that number is an old number, and that number comes from epidemiological research that only counted people that have been clinically diagnosed. They don't count all the others. And in fact, companies are now doing trials in a target population that is not clinically diagnosed. So those numbers don't apply. But there are recruitment difficulties, and those recruitment difficulties has to do with the inclusion-exclusion criteria. For some gene therapy trials, they are so strict that it is indeed difficult to find the people that meet those criteria. And of course, there are limited access to people that are in HDISS stage 2, early stage 2, and of course stage 0 and 1, but I'm not defending trials in stage 0 and 1 at the moment. So our new Enroll 2.0 will help with this aspect. One thing that people need to realize is, out of all clinical outcomes, not considering digital outcomes, total motor score is the most sensitive to change when we are speaking about HDISS stage 2. And people might say, and the CUHDRS? Yeah, the CUHDRS has been developed mostly with people in stage 3. When we compare CUHDRS in stage 2 with total motor score, there is very little difference in sensitivity. So this is a paper published by Doug Langman, where he made calculations for sample size trials using total motor score. And of course, this is good news and bad news, because most of these calculations shows that it's impossible to do a trial that is less than 2 years. Ideally, it should be 3. And the populations still are in the range of 500 up to 1,000, maybe 600. You can read the paper and notice the details. The important thing is we did similar calculations with Jim Mills and Jeff Long. In this case, the outcome is not total motor score, but is time to stage 3, so it's a time to event design. The issue here is we are enriching the population based in different PIN scores, beams of PIN score, but yet the message here is Even time to event or total motor score numbers are similar, and the order between 500 up to 1,000. Not a big difference. So again, these sample size calculations are made for this target population that is late stage 2 and maybe early stage 3, and that's where most of the companies are operating now, and probably rightly so. One important thing is everybody speaks about, oh, accelerated approval again. And by the way, the one that FDA did for ALS, because ALS is a neurodegenerative disease, because Tofferson is a ASO, there is a lot of commonalities. The issue is, of course, the biomarker in this case was NFL, and it changed in a very significant way. It changed more than 50%. It was not just twitching around the baseline. It really changed. But more importantly is that FDA says and that they triangulated the values of the biomarker with clinical endpoints at the same time, so cross-sectionally. So they were not just hoping that the clinical endpoint would be met in a later study, later after the approval based on the biomarker. There was already a relationship with clinical outcomes. And so if people, and all this is published, all this data is in the FDA website. So if you want to think seriously about accelerated approval, you have to think at least what will be the clinical outcomes that will be secondary in your trial, but that will be able to triangulate with the biomarker. So another issue that people speak a lot is about therapeutic window. And therapeutic window meaning two things. One is how late is late that you have so many secondary changes and you have so many comorbidities that it is too risky to operate there. And we don't know absolutely for sure, but I would say HDISS stage 3, after the very early stage 3, which is Schussen and Fan stage 1, it starts to be late, and it's probably to be avoided. The other issue is how early should we treat? And there is two differences, two important aspects to this. One is, if you are asking how early should you treat in clinical practice, in medical care, one day when there are drugs available, you would like to treat people as early as possible, provided the benefit risk of that intervention allows it. And it will be different benefit risks. For gene therapy that is intraparenchymal, the benefit risk will be less. If for, let's say, small molecules, the benefit risk is likely to be better. So you might come earlier and early in medical care. But you don't gain anything at the moment to do very early trials because there is nothing that you can measure. The sample size will be huge, and there is no good reason to do that except if you have a mechanism of action that is only operating in a narrow window. And if that is not the case, you are better doing a trial in a population that sees change. So, depending on mechanism of action and what we know about the disease, you might have to go early. But so far, there is no mechanism of action, maybe with the exception of excitotoxicity, that is supposed to have a narrow period of action. So, I would say, I don't have time to elaborate much of these aspects, but I would say I enumerated a number of challenges to do selection of population, outcomes, and duration of follow-up. There is a conservative solution for these problems, which is to do a classical parallel randomized controlled trial, that you target in that trial a population late in HDISS stage 2, You power the trial in caudate and putamen change, but you include key clinical outcomes, and you follow people for at least 2 years and ideally 3 years, and you don't expect FDA to agree to this today. You have to take it at risk. And this is conservative. For a daring solution, there are many possibilities of running adaptive trials, and there are many possibilities for adaptive trials that we'll have to discuss in another session. So another important challenge that we face, aside the ones I already mentioned, is the development of pharmacodynamic biomarkers and predictive biomarkers. The field have relied on mutant huntingtin in the CSF to prove target engagement for huntingtin lowering therapies, and also some programs on the blood. There are some issues still to be solved with mutant huntingtin assay, and there are difficulties with certain programs to get proper measurements, which are understandable. And so this issue of a pharmacodynamic biomarker, even for huntingtin-lowering programs, is not solved. And yesterday, during the somatic instability session, there was a lot of discussions about biomarkers for somatic instability targets. And for those, there is no sufficiently quick biomarker. Vanessa mentioned the blood somatic instability, and indeed, there is a good data on that, but the blood is very slow to change, and so this biomarker, although should be measured, will never help a company make a go, no-go decision, because it changes almost as fast as a clinical outcome. So you don't need that biomarker to make a clinical trial. So you should measure it to prove proof of concept and et cetera, but it does not solve your problem of a quick read. So it's an open problem that needs to be solved. And regarding predictive biomarkers, meaning biomarkers that can lead to the holy grail of reasonably like surrogate, these biomarkers, the best candidates we have at the moment is caudate atrophy and putamen atrophy, for which we have a reasonable amount of data. I know that everybody's praying for NFL. It's your God is very, very busy, because you are praying for accelerated approval. You are praying for NFL as a biomarker, et cetera. Just get real. And NFL still have a long time to go to be more than a safety biomarker. So the only thing I can say, we are amending a role. Yesterday, there was a very quickly run through the changes of Enroll 2.0. Probably the ones that have not read about them could not catch everything, but important things are coming along. We are making a much bigger investment in biomarkers, including in imaging. We are investing in the early population, so we are really trying to understand what is going on. And so we can support, as we have been doing, many of the trials and other studies. And that's it. Thank you. Questions? No, I take questions.
If you want, we can do questions at the end as well.
Yeah, that's fine.
All right. Well, thank you very much, Christina. We're going to have a panel at the end for questions. We have a really packed session, a very exciting session, 7 speakers. The first is Dr. Michael Hayden. He's a mentor of mine, a dear colleague, and more importantly, a dear friend. But for this talk, he's the CEO of Prelenia. Michael.
Okay. Thank you, Blair. And just want to say, this is a meeting that I love. This is a meeting where you get more hugs than any other meeting I go to. So it's really cool. And I just want to compliment HSG. The morning session was really very moving, very thoughtful, very intuitive, and I think all of us appreciated it. So I'm going to be talking to you, giving you a quick update on pridopidine and treatment of HD. And these are some of the disclaimers. I'm numerous things, but here I'm the CEO of Prelenia. There are 3 things I'm going to deal with today. An update on Proof HD, I'm now for the first time going to share you the data on the 2-year follow-up. This is the open label data. And then I'll show you some data on pridopidine and how its effects are not masked by lower doses of certain ADMs. Just to remind you, the target for pridopidine is the sigma-1 receptor. It's located on the mitochondrial-associated membrane of the endoplasmic reticulum, and it is able to reverse many of the changes that are seen contributing to HD as well as contributing to other neurodegenerative diseases. It restores calcium, decreases ER stress, restores calcium homeostasis, mitochondrial functions improved. The result is enhanced autophagy, improved synaptic function, BDNF is secreted, the spines and connectivity throughout the brain is improved, and dopamine is released. These mechanisms are shown and have been published and shown to be dependent on sigma-1. You delete sigma-1, you lose the effect completely. Just to remind you, of course, just a summary, in all these measures, there's progressive decline. All the patients here were mostly early ISS stage 3, and some a little later, but mostly early. And essentially, for all these measures I'm going to show to you, there's progressive decline. This is a summary of all these events and measures. We improve CHDRS. There's slowing decline of TFC. Stroop and other measures of cognition are improved. Fine measures of fine motor coordination really are improved, and quality of life, which I won't go through today, is preserved throughout. Here's just showing you CUHDRS. The data I'm presenting you initially is patients off ADMs, which was recognized as a potential confounder and pre-specified. What you can see here associated with this is that there's below the line is in favor of placebo, above the line in favor of pridopidine, and what you can see here is that this is all improved all the way out to week 78, and based on the paper by Schobel, who's going to be a later speaker, which is really, really important, what you can see here is an effect of 0.2 to 0.3 per year is defined as clinically efficacious, and essentially all these measures are at that level or greater. This is just showing you a summary of all the data. What you can see here, firstly, CUHDRS. Remarkably, you see an improvement initially, followed by progression of disease, but there's a slowing of decline all the way out to week 78. Stroop, again, as an indication of cognition, is improved, never goes below baseline. And again, fine motor, this is, what is that? Fine motor coordination, this is finger tapping, again, improvement, and then stays at baseline all the way out to week 78. Just to show you now the open label data, just providing more data. This is just showing you the persistence of effects for numerous measures. This is CUHDRS in TFC, and you can see while there is a decline, we also really wanted to compare this to a natural history control, and this was propensity weighted, and also for all the covariates seen in our study, and this is, we're grateful to Sarah Tabrizi, who gave us access to this database. And here you can see, compared to TRACK-HD, both for CHRS and TFC, these are really showing slowing of decline all the way out now to 2 years. And furthermore, this is just looking at cognition, one measure, Stroop, and also fine motor coordination, which is important for dressing, feeding yourself, going to the washroom, doing your shoes, brushing your teeth, important measures. You can see very little deterioration, and this is comparating what's seen in TRACK-HD. Of course, TRACK-HD also had Q-motor, so represented the best natural history cohort that we could astonish. So now just to the final issue, showing you some impacts on the ADMs. Important to know, pridopidine is a strong CYP2D6 inhibitor. We've known that for some time. And furthermore, all the regulatory labels for the VMAT inhibitors and many, not all, antipsychotics talk about dose reduction when given with CYP2D6 inhibitors. Furthermore, if you look, I know about this drug. I took it to approval together with the team at Teva. What you can see here under warnings What you do see is worsening in mood, cognition, rigidity, functional capacity, and difficult to distinguish between adverse reactions and progression of disease. When you actually look in terms of using this with CYP2D6 inhibitors, it gives specific recommendations. The exposure is greater, and it gives specific dosage recommendations associated with this. We actually did PK studies took patients who were on predopidine and Austedo, and predopidine and placebo and Austedo, and what you can see here just is showing you some of the PK levels. Essentially, what you can see is if you give predopidine together with deutetrabenazine or Austedo, you get an increase in exposure about twofold. So we were intrigued by this, and the question we asked ourselves is, do patients on lower doses of predopidine have an effect? Because now the exposure, and we know this at lower doses, would be around the regular dosage of this particular medication. What you can see here, just showing you again, this is the cUHDRS. And fortunately, in the study, we had some patients who were on low dose and some patients on regular or higher dose, and we were able to post-hoc look and see how they were doing in the study. And what you can see here, this is the low dose, smaller numbers, but these are following the dosage recommended in the labels. You can see essentially these patients on low dose or antipsychotics or antidopaminergic medications really show similar response when it's combined with predopidine. And when you go to the high dose, these patients, in fact, lose the response. The high dose masks the effects of predopidine. But there's numerous others. Just to give you also, we have done a survey now to hear the patient's voice. And this has been done with Canary. It's done externally. And Seth Rotberg will be presenting on this in much more detail. But the voice of the patients, these are independent interviews done, is that they recognize patients' improvement in cognition, reduction in caregiver burden, increased ability for activities of daily living, improved motivation, and certainly better quality of life. And these are just some quotes from patients where without medication, I felt more irritable, but now taking medication, I feel so much more calm, have seen benefits. Essentially, quality of life is excellent compared to siblings and their progressive disease. One of the things we didn't expect was the improvement, the reversal of symptoms, etc. So we will be putting the story together very formally to hear the voice of patients. And overall, we really are learning so much more about the drugs. These have implication not just for predopidine, but the use of antidopaminergics in other studies. A paper has just been accepted, which will outline some of the literature on this. And finally, let me just summarize. Benefits in HD on progression, cognition, motor in patients off ADMs. We see long-term sustained efficacy now up to 2 years. ADMs are not contraindicated with predopidine. They can be used, but the positive effects are not masked by the lower doses of certain ADMs. I just want to say there are 5 posters here that outline some of this, and these are some of the posters. We welcome you coming to this that gives much more detail around all of this. And I do want to say thank you to so many members of this community, the PIs, which were Andy Fagan and Ralph Freeman, the co-PIs, Sandra Kostick and Rasa. Sarah and Doug have provided ongoing support for access to track. The HSG has been a very important partner, and of course the HD patient advocacy groups and colleagues at Prelenia. Thank you all so much. How long was that? Oh, I'll go then.
Okay. So Michael and the other speakers will join me up at the desk after their talks for the question and answer session. So great, Michael is just under time. We're aiming for about 7 minutes, and at 10 minutes I'm going to stand up and loom. I didn't have to loom over Michael. I hope I won't be looming over anyone. Amy Lee Bredlau is going to now give her talk. She's the Senior Medical Director at PTC. Amy.
Thank you. I'm really happy to be here today speaking on behalf of PTC Therapeutics, and thank you for the invitation to speak. We'll do an update today on our clinical trial, Pivot HD. I do work for PTC. You won't be surprised to know. I do have good news to share with you from our perspective. We actually have a name now for PTC 518. We'll be calling it Votoplam for the rest of this talk. So it is a, Votoplam is a small molecule. It's taken orally once a day. It's titratable, it's reversible. It's a highly selective and specific target. Drug reduces HTT mRNA within just a few hours of being taken. It's delivered directly into the brain across the blood-brain barrier, and it's not effluxed. This allows us to keep our doses very low when giving to patients. It does also provide uniform lowering in the brain. We were able to look in our animal models to each area of the striatum to confirm that there was targeted drug delivery to all areas of relevance. But this being an oral molecule, it's actually delivered to the entire periphery, and we hope that that means that some peripheral symptoms might also be impacted. Pivot HD is a phase 2 randomized placebo-controlled study. It has completed enrollment, but and the participants on study, they're given one of 3 arms, 5 milligram, a 10 milligram, or a placebo arm. We shared a year ago the updates on our 12-week data, which did show HTT lowering. What we're going to talk about today is just a sneak peek at our 12-month data. It's the same subset of participants. There's 32 participants, the first 32 to enroll. So obviously, they were the first 32 to complete as well. And I think the most interesting and helpful bit of information that we've received from these patients is that there are no NFL spikes. This is the full 12 months on those. It's CSF and NFL, the full 12 months for those 32 participants. And for those of you who listened to my colleague Brian Beers talk yesterday, he also shared that there were, in the CSF, we've seen no spikes in CSF protein or leukocytes. We're really happy with that. Our key objectives for this interim peak at month 12 was to demonstrate durable protein lowering and durable safety, activity in biomarkers. And we're really happy to say that we have achieved that we only have a few minutes here. So you saw more details yesterday at Brian's talk if you attended that, but I don't have time to go through everything today in specific detail. But we're very happy that our biomarkers are consistently lowered by Votoplam in the CSF and the blood. We had signs of stabilization with TMS and cUHDRS in this small subset of participants. And again, they did well and tolerated this very well. And I want to thank all of our collaborators. We have sites all over the world and organizations such as HSG and CHDI, among others, that have really helped us to get this far. So thank you for your attention. I want to thank the participants in the study, their families, for all of their sacrifices and time and effort to get us this far. And we will continue to develop Votoplam, and we'll take it as far as the science allows.
Our next speaker will be Peter McColgan. He's the Global Development Leader at Roche. Peter?
It's great to be here today to be able to share an update about our tominersen program. So I first of all want to share some fluid biomarker data from Generation HD1 and put it into context of what it might mean for our phase 2 tominersen study, Generation HD2. I'll then provide an update about where we are in terms of recruitment. So first off, I'm a full-time employee of F. Hoffman-La Roche. And let me go through an outline of today's presentation. So I want to first explain what biomarkers in the CSF can tell us, so YKL-40, total tau, and GFAP. I then want to share an exposure response analysis from generation HD1 and understand what it means for generation HD2. And then I'll go through where we are with recruitment and the recent IDMC review that we've had on generation HD2. So what can these fluid biomarkers tell us? So let's first consider the cells in the brain. So we have a neuron, we have axons, microglia, and astrocytes. So if we think about YKL-40 first, this is expressed and secreted by microglia, and it can be considered as a measure of neuroinflammation. If we then consider total tau, this is a non-specific marker of neuronal death, and is somewhat similar in this way to neurofilament light or NFL. These are both non-specific markers of cell loss. p-tau181 is specific to aggregated tau, and GFAP is a measure of astrocytic reactivity. But importantly, what do these measures do in the natural course of Huntington's disease? So we know that from data from Sarah Tabrizi's group and Ed Wild's group, that these fluid biomarkers go up in HD relative to healthy controls. So we can think that maybe them going up is probably a bad thing, and potentially them coming down is a good thing. Okay, so let's look at some data from our phase 3 study, Generation HD1. So this is an exposure response analysis. Here we pull those on treatment. So if those of you may be familiar with the study, we had people on 120 milligrams every 8 weeks and 120 milligrams every 16 weeks. We can pull these individuals together and measure the level of tominersen in the CSF, and that's known as exposure. By doing this, we can then split individuals into quartiles, going from highest exposure to lowest exposure, and then compare with placebo. So here on the plot, we're looking at CSF NFL in purple and CSF mutant Huntington in pink. If you look at the highest exposure quartile, you can see that NFL goes up, it peaks at about week 21 and comes back down again. Mutant Huntington lowers, and it's around in the realm of about 50 or 60%. If we move down to the lowest exposure, you can see that we avoid the CSF NFL increase, but we still have mutant Huntington lowering in the realm of about 20 to 30%. So we avoid the unfavorable biomarker change, but we still have CSF mutant Huntington lowering. And the important thing is the lowest exposure quartile is the exposure that we're targeting with the doses in Generation HD2. And I'll come on to speak about this more in detail later. Okay, so let's look at CSF YKL-40. This is in pink. Again, you can see at the highest exposure, it goes up at week 21, comes back down again. At the lowest exposure, you avoid that increase. Then looking at CSF total tau and CSF p-tau181. Again, at the highest exposure, you can see it go up at week 21 and come back down again. But this time, it comes back down below baseline. If you go to lowest exposure, you can see that you avoid that increase, and it actually goes below baseline again. So this is potentially a good thing. So let's zoom in on the lowest exposure quartile now. Sorry, GFAP is the next one. Highest exposure GFAP, again, it goes up. Lowest exposure, you avoid that increase. So the same story at the lowest exposure. So we can now zoom in on the lowest exposure and look at what these biomarkers are relative to placebo. So Q1 here, blue is lowest exposure, gray is placebo. So with CSF mutant Huntington, you've got lowering of around 20 to 30%. CSF NFL, you can actually see that it trends below placebo. YKL-40, it goes below placebo. Total tau and p-tau181, again, it goes below placebo. GFAP, it goes up and then comes back in line with placebo. So putting all this together suggests that at the lowest exposure, akin to the doses we're targeting in Generation HD2, we avoid the unfavorable biomarker changes, and we potentially see some favorable biomarker changes. This is, of course, an exploratory analysis. It's post hoc, but it gives us some indication of where we think we might be with the doses in the new study. So just to summarize, CSF YKL-40, total tau, and GFAP can tell us about neuroinflammation, neuronal death, and astrocytic reactivity. When we look at this data in Generation HD1, we can see that at the lowest exposure that we're targeting in the new study, we avoid the unfavorable biomarker changes. We potentially see some favorable biomarker changes. Okay, great. So let me now switch gears and give an update about where we are with recruitment and the recent IDMC review. So I'll start with just a brief overview of Generation HD1. This is a phase 2 study for tominersen, looking at individuals with prodromal or early manifest HD. We're looking at 100 milligrams every 16 weeks, 60 milligrams every 16 weeks in placebo, 16 months of treatment with a common close. So that means that once you get to 16 months, you continue on blinded treatment until the last participant has had their last visit. So let me just outline the IDMC. So we have an independent group of scientists, independent from Roche, that are looking at the data every 4 to 6 months. This is made up of a statistician, a brain imaging scientist, and 3 expert Huntington's disease clinicians. So the role of the IDMC is to review safety and clinical outcomes and review the balance of risk and benefit. The IDMC look at safety, clinical, brain imaging, and plasma NFL every 4 to 6 months. So the IDMC can make a recommendation to either continue, modify, or stop the study. And I'm delighted to say that at the last IDMC review, where they looked at unblinded safety, clinical, MRI, and plasma NFL data, in 219 participants, up to 5 doses, 166 days on treatment, they recommended us to continue as planned. So this is really reassuring, and of course these reviews will continue. The other great news is that we're now more than 80% recruited, and this is really thanks to all of you guys, the community, the PIs at site, the site staff. It's really a milestone because it's taken us some time to get here, but we're more than 80% recruited, and we're going to close recruitment at the end of the year. So with that, I'd like to say a massive thank you to everyone that helps us along the way, most importantly, patients and families, but also all of the HD clinicians at site and all the site staff, because without you, we couldn't be where we are today. So thank you so much.
Thank you, Peter. Great timing. And Jane Atkins, the program lead from in Portfolio Strategy at Wavelight Science, is our next speaker. Jane? Oh, there we are. Great.
Good morning. I'd like to thank the organizers and HSG for giving me the opportunity to present today the results of our Select HD study. I'm going to be making forward-looking statements, and I'm an employee of Wave Life Sciences. So in the next 7 minutes or so, my goal is to leave you with 4 key takeaway messages. The first is that WV003 selectively and durably knocks down mutant huntingtin in the CSF to a maximum level of 46%. This exceeded our predefined criteria of 30%. Secondly, WV003 did this in a allele-selective manner, sparing wild-type protein. And the 30 mg Q8-week dosing regimen was generally safe and well-tolerated. These 3 points taken together warrant further clinical development of 003. Lastly, we show for the first time a demonstration of an association between the amount of mutant huntingtin lowering and the slowing of caudate atrophy. So WV003 is a first-in-class investigational oligonucleotide that's allele-specific and targets mutant huntingtin. It does this by targeting a single nucleotide polymorphism, or a SNP, that's associated with a mutant huntingtin allele. And we call this SNP SNP3. 003 works at the level of the transcript to prevent the production of mutant huntingtin protein. Because it's allele selective, it spares the wild-type allele, enabling expression of wild-type protein, which we know is critically important in CNS and neuronal function. 003 is the most advanced allele-selective molecule in the clinic today. Turning now to Select HD and the study design. Select HD was an adaptive phase 1b/2a study that was designed to rapidly assess 003. It had 2 parts, a single ascending dose phase and a multi-dose phase. The single ascending dose phase looked at 3 different dose levels, 30, 60, and 90 mg, and the multi-dose phase looked at 3 doses of 30 mg given 8 weeks apart. The primary objective was safety and tolerability, and we also looked at exploratory CSF biomarkers, mutant huntingtin, wild-type huntingtin, and NFL. We also looked at clinical assessments and volumetric MRI on an exploratory basis. Because of the limited time that I have today, I'm going to be focusing the rest of the talk primarily on the multi-dose phase results. So baseline characteristics were generally balanced across all cohorts. Multiple doses of 30 mg of 003 was generally safe and well tolerated. AEs were balanced between the placebo and the treated group, all of which were mild to moderate in intensity. We did see a slight imbalance in treatment-related AEs that were mild to moderate in intensity. Mild events included hyperreflexia, and moderate events included CSF biomarker lab abnormalities, including CSF protein elevations. At the time of the data cutoff, there were no SAEs. So ventricular volume has emerged as an important safety measure within HD. And when we looked at ventricular volume changes, we found that they were consistent with natural history, and we saw no cases of hydrocephalus in the study. So turning now to the target engagement data. This slide shows CSF mutant huntingtin levels and wild-type huntingtin levels as a function of study day. On the left, you can see that we saw a maximum amount of mutant huntingtin lowering of 46% at day 169 of the study. This effect was durable, as on day 197, lowering was still 44%. We saw consistent preservation of wild-type huntingtin protein compared to placebo and baseline throughout the study. This is the first time that this has been observed in the clinic and confirms WV003's intended mechanism of action. We were super excited to see this. Basically, it works. Yay. So these data, particularly the durability of 003 that we see here, combined with additional PK/PD modeling, support moving forward with a less frequent dosing regime going forward. We're going to be looking at Q12 weeks versus Q8 weeks. So we also looked at NFL levels. This is a graph of relative CSF NFL levels as a function of study day. This is a spaghetti plot with each line showing an individual patient's NFL profile. The gray box delineates the 5th and the 95th percentiles of the NFL levels that were seen in placebo patients. So a couple of things. The majority of patients' NFL levels, treated patients, were within the placebo range. We did see some increases in 5 patients, only 2 of which were still increasing at the end of the study. We did further analysis on this and found that these NFL levels, there was no correlation with the number or the intensity of AEs, and we also found that it didn't correlate with changes in caudate volume. We then looked at plasma NFL, and here we saw no significant difference between treated and placebo. To our knowledge, this is the first time that there's been this disconnect of the plasma NFL profile and the CSF NFL profile. Usually they tend to track. We're not sure what this means, but it is something that further differentiates 003 from other molecules. So SELECT-HD was not powered to look at significant clinical outcomes or MRI, but we did see some observations in the exploratory measures that help inform 003. So here we looked at caudate atrophy, or caudate volume, changes in caudate volume. And when we looked at after day 169, that's the last MRI that was taken in the study, we were encouraged to see that the change in caudate volume for treated patients was slightly less at 4.68% compared to those in the placebo group of 5.1%. Moreover, what we were very excited to see was that we saw a significant correlation between the amount of mutant huntingtin lowering and the change in caudate volume. Basically, the more mutant huntingtin lowering we saw, the less loss in caudate was observed. And we saw a similar correlation with 003's CSF concentration. As planned, we looked at clinical outcomes, cUHDRS, SDMT, TMS, and TFC. We were not surprised to see no clinically significant difference between any of these measures in the study out to 6 months. Importantly, we also saw no worsening in any of these markers. On TMS, we were encouraged to see a point estimate difference that favored 003. This is not statistically significant, but this change of approximately 4 points is greater than the minimally clinically important difference of around 1.8 to 2 points that you would expect to see in 6 months. So this is encouraging. Again, N small, but encouraging. Right. So to summarize, 4 points again. 003 selectively and durably knocks down mutant huntingtin to a maximum level of 46%, and it does this in an allele-selective manner. The safety and tolerability profile is encouraging, and we intend to move forward to assess a Q12-week dosing regimen versus a Q8-week regimen. We have seen for the first time a correlation between mutant huntingtin lowering and the slowing of caudate atrophy, and we're super excited to see this. Right now, we are in the process of getting regulatory feedback, and we expect to have that by the end of the year to inform next steps on this program. So lastly, I'd like to thank the investigators and the sites, in addition to, as well as the patients and their families, all who were involved in this study. Couldn't have done it without you. Thank you.
Thank you, Jane. Our next speaker is Scott Schobel. He's the Chief Medical Officer for Vico Therapeutics. Scott?
Good morning. On behalf of my colleague Micah Mackison, our CEO, and I, the two representatives of Vico at the meeting, a small company, I'm very pleased to share with you a trial update on VO659. As Blair said, I'm the Chief Medical Officer of Vico and own stock. VO659 targets directly the CAG repeat motif, which is, from many lines of evidence, thought to be causal in the pathophysiological cascade of Huntington's disease. It does so in a way that is a twist on an ASO mechanism. It sterically binds to the motif. To the extent that the motif is longer, more molecules of VO659 bind. The result of that differential engagement of the molecule on the extended CAG repeat expansion is to, in a cellular context, have more of an effect the longer the CAG repeat. And that's shown from cellular results that we've generated on the right-hand panel. By definition, if it's not already obvious, this is an exon 1 targeting approach as well, which we term allele preferential in nature. So, on to the update. We have an ongoing phase 1/2A clinical study in 3 individuals who have CAG repeat expansion disease. These individual patient groups include Huntington's disease participants who we've begun to enroll at the 40 milligram dose level. In addition, we've enrolled SCA3 and SCA1 participants who are also in the family of the polyQ diseases. We started off in SCA3 to be efficient, to escalate to the 3rd dose level, and then we also expanded enrollment to include the HD and the SCA1 participants. Today, I'm pleased to share with you safety tolerability data to date from all participant groups. And then we have mutant huntingtin protein and neurofilament light protein information from CSF, which we've recently presented at EHD and through day 85. We've recently extended this to day 120, and I'll show that new today. We also have a comprehensive presentation. Well, actually, it's a 5-minute presentation at ICAR upcoming. It's very short, but essentially, we'll include more SCA1 and SCA3 data there next week. On to the baseline demographics and clinical characteristics of the HD participants. Here are the 6, balanced for gender, CAG repeat lengths representative, TMS scores mainly representative of early stage HD, with the exception of 2 individuals who had more prodromal range. We did not use a symptom-based threshold for enrollment, so these last 2 individuals who had TMS scores of 5, in fact, had below limit of quantification measures of CSF mutant huntingtin, and that's shown on the right panel. The remainder did have measurable levels of CSF mutant huntingtin, and therefore these 4 individuals could be used for post-baseline analysis. And so when we looked at the post-baseline analysis, originally through to day 85, from our first interim, we could see a 28% mean reduction at day 85, with the effect having an immediate onset at day 29. This implies that we could probably, in all likelihood, should back off frequency. This is a 4-dose phase 1 study separated by 28 days, but since we have an effect after the first dose, in all likelihood, we could back off frequency to something much more participant and individual friendly. And what we have in mind is the range of every 4 to 6 months or even less frequent. So we were pleased to see this. And then also from the first interim, we saw the NFL time course. And essentially, this time course is more or less bouncing around 0, slight increase in magnitude in the early part of the time course. It's on a downward slope to day 85 at 5% increase at day 85. We take this to mean no evidence of sustained increase or decrease in the measure from this rather limited data, but nonetheless, that's the finding. So the new information includes the change from baseline to day 120 analysis. We wanted to see what happened over further time courses, and that's shown here on the left for CSF mutant Huntington and on the right for CSF NFL. What we can see is the magnitude increases to day 120. We're now sitting at about a 38% reduction at day 120 in the available participants from this analysis. NFL, we've just slipped below baseline. We're at negative 2.3%, with some individuals going lower, as shown by the confidence band. So we are pleased to see an extension of the previously observed first interim analysis results here at day 120. On to safety and tolerability from all participants. This is an adverse event summary that's current as of yesterday. For full transparency and wanting to keep the community informed. So what we have here is what we've previously shown, which is an unchanged profile for the HD participants from EHDN. We've had a total of 2 serious adverse events. One was non-drug related, a worsening of depression in an individual. The second was lumbosacral radiculitis, which is a not commonly but observable effect with CNS intrathecal bolus ASO exposure. It's inflammation of the spinal nerve roots. And then when we look across the other disease groups, we see the same lumbosacral radiculitis at the 40 milligram level in 1 SCA3 participant and 2 SCA1 participants. The clinical course of these is such that the onset was after 3 or 4 doses. We believe it's related to cumulative exposure of the ASO. And 3 of the 4 cases for which we have close follow-up data are showing signs of reversibility, which is also consistent with the field's recognition that ASOs themselves, when you stop drug exposure, are reversible in terms of their effects generally. And that's what we observe for these 3 of 4 cases that were the first cases we've had. And we're well into the post-dose follow-up period now, several months. So I think this is probably what we're going to see for this first phase 1 study. In terms of adverse events by preferred term, we can see commonly observed adverse events for intrathecal bolus ASO, headache. We do see CSF protein increase consistent with the pro-inflammatory nature of ASOs, dizziness, fall, nausea, and post-lumbar puncture syndrome. So in summary, VO659 is the first CAG repeat targeting allele preferential ASO to demonstrate CSF mutant Huntington lowering in HD. We're on a downward trajectory, clearly from available data. We believe that the onset of this effect supports a less frequent, no-load dosing regimen. We clearly see no change in CSF NFL over the early time course, and it's also on a downward slope through available follow-up. We do see the drug generally to be safe and well tolerated, but not entirely owing to the lumbosacral radiculitis cases. However, we know how to dose around this. Essentially, programs that have seen this, what they do is they drop frequency primarily, and then the pro-inflammatory nature reduces, and the problem basically goes away. So that's fully what we intend to do on our program going forward. We're really cautiously optimistic about the potential of VO659 in HD. Clearly, we need to be careful and take all the lessons of the field studying this forward. So I'll conclude by just providing a brief program update. So our phase 1/2A study is still ongoing. We're entertaining modeling an ultra-infrequent dose approach, which we're in discussions with our IDMC, who has endorsed our proposal to test an ultra-infrequent approach in a last cohort of the phase 1/2A study. Nothing set in stone. We'll update you when we have more clarity. In addition, we're planning a phase 2 study to explore effects of the infrequent no-load approach. using a low dose range, given the apparent potency of VO659. We think we can play at 40 mg and below for chronic dosing, and that's really based upon all of the convergent evidence and the anticipated long half-life in tissue that we see, looking towards once to twice a year dosing if possible. We're in discussions with regulators for the next steps, and I want to just point out the below causal pathway. We believe that it's a preponderance of evidence approach to prove any clinical effect of a drug, and that it may not be one of these nodes in particular, but it may be a combination of all of them going in the same direction that would give us confidence that we have a drug which has putative benefit risk. And that's what we will be going for as we develop this drug further forward. So I just want to finish with a note of thanks. What a great meeting, what a great community, so much innovation. It's just a real privilege to be a part of this. And thanks. Without this group, no progress can be made, for sure. So thanks.
Thank you, Scott. So our next speaker is David Margolin. He is the VP of Clinical Development at Unicure, and I'll invite David up to the stage.
Advancing the slides? We'll see. Yes. Hi, I'm an employee of Unicure, and will be introducing today AMT-130, an investigational gene therapy for Huntington's disease. One-time administration leads to a treatment effect with potentially long-term effects. The drug works through the mechanism of RNA interference, about which you might have heard yesterday, leading to suppression of production of mutant huntingtin, including the highly toxic exon 1 splice isoform, both mutant and wild-type huntingtin affected by the treatment. The drug is administered by direct injection into the striatum, as illustrated in the cartoon on the right. But axonal transport carries the therapeutic to also cerebral cortex and other diseased areas of the brain. AMT-130 has been in development program, includes 2 clinical studies, one based in the US and the other in Europe. At this meeting last year, I presented an interim analysis of only the US-based study. Today, we're able to provide data pooled across both studies and offer additional follow-up. We have, of the 29 subjects who've received the treatment in the cohorts 1 and 2, we have 2-year follow-up data on 21 of those subjects. This introduces the study design, which was quite similar across 2 studies. They had, in fact, the same eligibility criteria, dosing, and follow-up procedures. We've completed dosing in cohorts 1 and 2, the data I'll be presenting today. Cohort 1 received a low dose of AMT-130. Cohort 2 received a 10-fold higher dose. There is a cohort 3 that's currently enrolling, but we don't yet have the efficacy results from that cohort. The US-based study also included a randomized sham surgery control group with 1 year of follow-up before unblinding and a potential crossover to receive active treatment. The safety results are summarized here. The neurosurgical administration of the therapeutic sometimes led to serious adverse events. There were no drug-related SAEs on the low-dose arm. The high-dose arm did have several serious adverse events, which we believe were attributable to a CNS inflammation triggered by the AAV capsid that carries the therapeutic into the brain. At the meeting last year, I presented the interim analysis relative to a comparator group drawn from the TRACK-HD and TRACK-ON studies. This year, we have a newly expanded natural history cohort, drawn again from TRACK-HD and TRACK-ON, but with additional subjects, also from PREDICT-HD. All subjects had to meet the AMT-130 study eligibility criteria and have at least 2 years of follow-up data to match the 2-year time point that we'll be examining. With those conditions, The external comparator control cohort included 154 subjects. Comparisons of the AMT-130 with those historical control data used a statistical method called propensity score weighting. It's a widely used statistical method that's accepted by regulatory authorities. The propensity score weighted comparator group has baseline characteristics that were very well matched to the patients who received AMT-130 as shown here. Top-line efficacy results are summarized here. The external comparator group, shown in orange, the propensity score weighted external natural history control, over 2 years showed a 1-point worsening on the composite UHDRS scale. Patients receiving AMT-130 had smaller changes. The high-dose group, shown in blue, performed best. Statistical comparison of the high-dose group and the pooled dose groups with the external control had p-values less than 0.05. This suggests dose-dependent slowing of disease progression after treatment with AMT-130. We also examined at the individual subject level whether the changes observed in cUHDRS and also in the TFC, the total functional capacity, were clinically meaningful. To do that, we used the threshold for the minimal clinically important difference published last year by Hamilton et al. From the CHDI and other group. And using those standards and thresholds, AMT-130 reduced the odds of clinically meaningful worsening by more than 40% for both of these outcome measures. In addition to the clinical outcomes, we examined CSF NfL concentration as a biomarker of the response to treatment. In natural history studies, notably the HD-CSF study, they reported that the CSF NfL level typically increases by about 10 to 15% per year. We performed an analysis looking at the total population of the HD-CSF study, selecting individuals who also met the AMT-130 study eligibility criteria, an N of 19, and found that consistent with the overall group, that selected subgroup had a 26% increase in the CSF NfL level over 2 years of follow-up. In our study, at 2 years, the pooled dose group had, in fact, a decrease relative to their own baseline, which was statistically significant, a 10, 11-point decrease in the CSF NfL level, consistent, we think, with disease modification, slowing of neurodegeneration. So summarizing, AMT-130, we feel has a manageable safety profile. The cUHDRS scores suggest that treatment with AMT-130 provides a clinically meaningful slowing of HD disease progression compared with this well-matched natural history control. And the CSF NfL levels provide an objective measure, with the result finding CSF NfL levels below baseline, suggesting a reduction or a slowing in neurodegeneration consistent with disease modification by the treatment. So our next steps going from here will continue follow-up of the cohorts 1 and 2, expected follow-up through 5 years from treatment. We're going to complete enrollment of the cohort 3 to investigate immune effects on perioperative safety. We'll be interacting with the regulatory agencies to define potential registrational pathway for AMT-130. Notably, this year, FDA granted AMT-130 a special designation called RMAT, Regenerative Medicine Advanced Therapies designation. This is a first for HD therapies and allows potentially for close collaboration with the agency, offering all the benefits of the fast track designation or breakthrough therapy designation programs. So we're looking forward to those discussions and continued data coming in from these studies. Thank you.
Great, thank you. Our next speaker, Silvia Panagoni, is the Chief Executive Officer at Som Biotech. Silvia.
Is this one? Okay, here we are. Sorry.
Okay, thank you. I'm the last one before lunch, so I will try to be very short. I'm honored to be here, and today I'm going to present the top-line results of our phase 2b study with our lead product to treat chorea in Huntington. I'm Silvia Panigone. I'm the new CEO of Som Biotech because I just joined recently. So who we are, just a snapshot. We are a European-based company based in Barcelona who focus on the development of drugs for the CNS and rare disorders, founded in 2010. I can say that has been pioneer in applying a proprietary algorithm artificial intelligence platform for the drug discovery, especially for the identification of new mechanism of action of all drugs. And recently, we really refreshed the board and the leadership team to prepare to the next level growth. The company brought already 2 products up to a successful phase 2b. I don't have to tell you what is the medical need of Huntington disorder, but I would like to underline that all the previous talk were related to drugs to treat Huntington. In our case, we are treating chorea symptom in Huntington. And regarding chorea, there are 3 drugs on the market, as you know, in the US, just 1 in Europe, and they are all VMAT2 inhibitors that, despite they can manage pretty well, I have to say, chorea. They all have black box warning for safety, mainly risk of worsening depression and suicidality. But on top of that, because this morning we talked about quality of life, they have additional concerns related to cognitive impairment, sedation, fatigue, akathisia. And so there's still a high medical need, especially for continuous long-term treatment. And this is where our drug is going to be hopefully position. Our lead asset is a new typical VMAT2 inhibitor. So we run our AI platform using tetrabenazine, a reference compound, and we found that bevantolol, a pretty old drug that was used for 14 years for the treatment of hypertension, show a very peculiar profile and can be used extremely successfully for the treatment of chorea and other disorder like tardive dyskinesia, for example. Bevantolol has been approved in a few European countries and in few Asian countries, now in the market only in few Asian countries. It is a beta blocker, so with a strong and long history of safety profile, with no history of worsening depression, no suicidal ideation of cognitive impairment. We define as atypical VMAT2 inhibitors, and we found the new mechanism of action of this drug that has completely different chemical structure from the tetrabenazine and the others. It still inhibits VMAT2, but inhibiting a different binding site. And on top of that, what is more importantly is that it binds and inhibits VMAT1. So we have a preclinical study ongoing with the University of Norway in Bergen, and hopefully there will be a publication next year. Because the rationale behind the different safety profile and efficacy profile may be exactly in the completely different mechanism of action of this drug. So the company ran a short proof of concept study for the first time in this population. The data has been published in 2023. Was a short 32 patients crossover study testing 2 doses of this drug that is given orally twice a day. The results were pretty comfortable, and satisfactory. The drug was extremely safe, as expected, and the efficacy and shows some significant improvement in chorea score. Upon a meeting with the FDA and with EMA scientific advice, we then decided to run a phase 2b study that I'm going to present today. So this is the study design. So it is randomized, double-blinded, placebo-controlled study assessing 2 doses of SOM 3355, 200 mg twice a day, so 400, and 600 overall, so 300 mg twice a day against placebo. For 10 weeks treatment, a maximum 12 weeks treatment, the primary endpoint is is evaluated as mean between the visit 4 and the visit 5. After the visit 5, the patients started the down titration of this product. These are the demographic and baseline characteristics, and just a couple of points which are extremely important, especially to interpret then the data. We can see that the three arms are pretty balanced in terms of characteristics of the patients and in terms of number. However, and this is not working in our favor to see the efficacy of a drug, there was a handful of patients taking neuroleptics as co-medication that were allowed to be included. Overall 12%, so fortunately not so high, and well balanced between the three arms. On top of that, we can see, and you can appreciate that the population, there is a pretty important part of the population included that were mild, calculated as CGI severity at baseline. More importantly, if you look at the TMC score at the baseline, we have pretty high variability, and some of that went down to 8.29. So I have to say, not working in the best condition to see a great drug effect. So this is at the first instance what we got. What surprised us is the pretty high placebo effect that we have seen, about twice what was computed for the study. The study was run only in Europe, these studies, in 7 European countries. And while in the overall population we did not see a statistically significant difference, if you, in a pre-defined sensitivity analysis, just taking out the handful of patients with neuroleptics that were expectedly co-founded the action of the drug, we immediately saw a statistically significant difference with the 300 mg. More importantly, if we consider the one with a stable baseline, more than TMC more than 12, we can immediately find, and not taking out neuroleptics, which reduced the number to 57, we see a statistically significant difference again with 57 only patients with the 300 mg, and the difference versus the baseline is even comparable to the marketed drug. Just one thing to notice, I mentioned that the primary endpoint, that the endpoint was calculated as mean between V4 and V5. You can see here that we realized that patients after the V4 started already down titrated, unfortunately, the 300. In fact, at visit 4, the difference from baseline is 4.53, so it's even more, which is pretty positive with rooms for improvement. Looking at the CGI is even better, and is not considering even the huge TMC variability that we have seen. We see a statistically significant difference. 2/3 of the patients show a benefit with the drug. Clearly here at the V5, you can see that for the 300 mg, the patients started already down titrated. Safety has been excellent, I have to say, as expected on the basis of the mechanism of action of the drug. So no somnolence, no akathisia, no fatigue, increased risk of fall, no sign of worsening depression. In a few cases, even slightly better, but just in a few cases. In terms of related treatment emergence, adverse event, we have just very few bradycardia. One was in placebo. One syncope, one hypertension, a suicidal ideation in the placebo group. Regarding the heart rate, which of course is a beta blocker, was as expected and even dose-related and within the normal range, apart the 3 cases of bradycardia, and returned to baseline after discontinuation. In terms of serious adverse event on the entire study, we had just 3. One was pneumonia. The other one was syncope after overdosing, and then we know a suicidal ideation with the placebo, that was in the placebo. So the summary of this top-line phase 2b data. By the way, we are still collecting the remaining data and some additional PK to evaluate better the drug exposure. Is with 300 milligram twice a day, show a statistically significant difference from placebo at the end of maintenance in predefined sensitivity analysis, excluding patients taking neuroleptics. Of those patients, the one with a baseline that with TMC score more than 12, or I have to say more stable, the 300 mg shows still a statistical significance and a difference from baseline that is similar to the marketed drug. The safety is extremely good, no worsening of depression again, no somnolence, no akathisia, which is extremely important, no fatigue, and no incidence of falls. So from this data, you have seen that there is room for optimization even regarding just the population and the study. We are pretty confident in moving forward with a pivotal study that will be run also, or maybe most even in the main part in the US, to position our drug, especially for a chronic continuous treatment of chorea in Huntington. So I thank you, all of you, and thanks, Blair. I hope I've been within the...
I'd like to invite all the speakers up to the stage. We have some time for questions. And I'll open it up to the floor for any potential questions. Please raise your hand, have a microphone. There's one over here. And I don't see any on the app, so we're going to go to the floor for questions.
Hi, I think this one's for Wave. I don't know if you can see. I actually have two questions, if that's all right, allowed. But I wanted to ask what platform you use to measure neurofilament in CSF and blood first, and then how you detect wild-type Huntington specifically and quantify it.
Okay, so I don't have specifics on the NFL.
Use the mic.
Oh, sorry. I don't have specifics on the NFL assay that we use, but it is qualified and validated for clinical use. In terms of the wild-type Huntington assay, we worked actually with CHDI to develop a wild-type Huntington assay. Again, I can go into the details. I don't think I want to go into that, but it's also qualified and validated for use in the clinic.
It's just my understanding is you can't actually target, at least through immunoassay and protein quantification, you can't specifically directly measure wild type over mutant. So it's just, yeah.
Yeah, all I can say is we have qualified and validated the assay.
Just to follow up, I think it's a depletion-based assay.
Yes,
Yeah. So I mean,
yes.
I think you can probably provide at least the basis for it.
Yes. Yeah, so again, I'm trying to go back in my head to remember all the technical details of it, but we do deplete first the mutant, and then I apologize, I am not up to speed on all the technical assays. We have posters on this and we published on it. So actually, if you want to come out, talk to me afterwards, I can dig into that more with you.
Just following up on that, I was interested in it looked like the wild type was going up.
Yes,
Can you
yes.
comment on
We
that?
did find that very interesting. So we have some hypotheses. It's known that, mutant Huntington, one of the things that it does, it forms aggregates, and in the process sequesters not only NFL, but also wild-type Huntington. So it's feasible that when we deplete mutant Huntington, that we're releasing those aggregates and potentially releasing wild-type Huntington. That's hypothesis.
Other questions? There we are. Please raise your hand and use the mic. Coming behind you.
Oh, thank you.
The question is for Unicure. Thank you all for your presentations. So I understand the FDA really pushes for sham controls early on, and I just wanted to hear your insight about the value of your sham arm on your initial study.
We had extensive discussion with FDA before launching the study, and they did push us to include a sham arm, and there was a randomized sham control for the cohorts 1 and 2. The key point of difference was over the duration of follow-up. We were concerned after hearing from the HG community and patients that if they were randomized, committing to a study in which they'd have to remain blinded, potentially on a placebo essentially, for 2 and 3 years, they wouldn't want to be in that study. The compromise was to say, okay, let's follow you for 1 year and then offer crossover. So at 1 year, however, and we presented these data last year and in other forums, the comparator group hadn't really worsened much. Their cUHDRS and TMS was basically still at baseline. So it turned out not to be very informative, except as a reference arm for safety events. In discussions around potential phase 3 or confirmatory studies, I think this will come up again, and we'll see where we end up.
Thank you so much. Yeah,
One of my colleagues has a comment. Did you have a follow-up?
No, I was just saying I would hope the FDA would consider this information on the really push for sham controls early on for Huntington's.
Can we ask questions too?
Absolutely.
So I was really intrigued with the Unicure data and WAVE data, because when you looked at the slides and the actual TFC, both were below. In your data, they were doing worse than placebo. Even though cUHDRS was somewhat improved, TFC was below placebo in those endpoints, and I wonder if you could comment on them. Worse than placebo. The one line was, they were both below.
So I'll just say, in our study, it's small n still, and it's only 6 months, and they were overlapping confidence intervals, so I don't think we can read too much into those results right now.
Yeah, and as I said, the comparator group at 12 months hadn't worsened. In our longitudinal follow-up, largely the subjects did better in year 2. We think that the neurosurgical procedure to administer the drug is a major traumatic brain injury, essentially, and it takes some time to recover. So in our analyses to date at 24 months, the TFC looks to be quite stable and improved relative to the comparator group.
We have time for one more question. Kevin?
Sure, thanks. Great talks, everyone. A question maybe for all 3 of the antisense platform organizations. We're seeing a consistent theme, right, where in all these programs, there's a need to back off the dosing frequency to sort of tune the safety profile and make sure we don't, I guess, reach too high exposures. Maybe, Peter, to start with you, what do you think we can learn from any of your programs about the safety of Huntington lowering and the extent versus the safety of the molecule itself? The challenge is interconnected. When you back off the exposure, you have less Huntington lowering. I'd just like to hear your opinions, recognizing we don't know.
Yeah, I think it's really difficult to disentangle the effects that you see from an ASO from the effects you see from lowering both mutant and wild type. And I mean, because these things are dose dependent, you just can't disentangle this. And I mean, I would love to know myself, because it's a question that we've had, ever since I've joined Roche, but I don't think we can answer it with the trial data that we have. I would love if there was even a preclinical way we could address this, but not with the clinical trials.
We're sitting at about 3 to 7 nanograms per ml on exposure at 40. That's kind of equivalent to the Roche high exposure band, if I understood the data correctly. With VO659, we get lowering in an NFL flat profile at that exposure. We do get some CSF protein rise, as I presented. I'd like to just say that I think no ASO that's been clinically efficacious has ever been stopped for a pro-inflammatory signal. Nusinersen is approved, causes CSF protein rise. Tofersen causes even more of a protein rise given monthly, over a gram, I think, on average. So I do think it's an important issue, but it's ultimately a nuisance issue, and it should be in the context of the mechanism of action of the drug. I think mechanism is going to be 1,000 times more important than nuisance pro-inflammatory events, which are avoidable indeed by backing off your exposure paradigm. And I think the key point is, can you get on, as you've suggested, can you get on-target effects still while backing off? And I think the tominersen data suggests that's the case. I'd be confident in VO659's ability based upon the day 29. But this is why we develop drugs, to optimize benefit risk. Yeah.
I really don't have much more to add. I think both of you guys made great points, and they would have been the same points that I would have made.
Great. Well, that's been a fantastic session. Thank you, speakers, for keeping the time, and join me in thanking everyone.